Well, I'm delighted to be here, and interestingly, this opportunity prompted me to rethink some of our own work. So I'm going to actually be presenting some analyses of our own research, because truthfully, in stroke and dementia, people present to us as human beings with deficits, with tragedies, with sudden onset or gradual decline, and we're not immediately thinking of whether they're a man or a woman. But it turns out that there are very interesting differences. And what I'm going to tell you about is how much more complex it may be or may seem than from what you first imagine. So I'm going to acknowledge the assistance of some of my graduate students and postdocs in helping to prepare and get ready some data from our own research. So it turns out that, brain aging and dementia, even stroke, is a very complex topic, and it's actually much more poorly understood than perhaps I even realized before I started preparing for this talk. So thank you, Lynn, for prompting me to do so. There are many factors: genetic factors, hormones, immune reactivity, of course, social and environmental, so biological but also social and environmental factors, all of which play a role. And it turns out that we can now measure the brain in a whole new way because we have computerized methods, right? So we can make measurements of the brain that we never could do even 10, 15 years ago. And so it opens the way to understand how the brain is changing over time and how dementia affects men and women differently. I think the issue of whether you do animal models, and some people might not like the idea of animal models, but we learn a lot from them in male rats or women rats, is another issue. And I think there's a great deal of need to do the women rats, even though they have that kind of stuff called estrogen, right, that makes it difficult. In most diseases, we're dealing with men or women, and actually men get breast cancer, believe it or not, but nothing like as often. Women never get prostate cancer, so that's one major difference. But in almost every other organ of the body, you're not immune because you're one sex or the other. And what we have to do is understand the differences in how they express themselves and why and why it's happening, because maybe we then can customize treatment better, we can anticipate, we can plan better. But the truth is we're still, I wouldn't say in the Middle Ages, maybe we're beginning to hit the Renaissance, in that people are now understanding that you've got to look at the differences in men and women and how they experience these diseases. I'm not going to be talking about, there's so many differences in men and women, mood disorder or women as caregivers. Men also are caregivers. They can be wonderful caregivers, but the vast majority of caregivers are women. So dementia, for example, not only affects perhaps more women, although that may vary in different cultures or different societies, but basically both men and women are being affected either as caregivers or as sufferers of these diseases. And generally speaking, women win the battle of living longer and also getting dementia more. So we will talk a little bit about that. So we have to proceed carefully to do these measurements well and to really understand and not be too lobby-minded to start with. Let's look at the facts. So I'm here to tell you today a little bit about the facts, and even I was a bit surprised by some of the results. First of all, there's not enough out there in the literature. There's a huge opportunity to do more research, which is why the Canadian Consortium for Neurodegeneration and Aging is important. And then the Ontario Brain Institute, which we can talk about afterwards in the question period, there's a huge opportunity to look at these differences as well, as a number of cohorts of individuals will be followed over several years from children to dementia. So we need more research. And I'll just go over some of these numbers. So in 1900, life expectancy was 47 years old. That was the average. There were about 3 million people in the US, 4% of the population. I don't think we have Canadian figures on this. About 4% were older than 65, and often they were not well. By 1990, the life expectancy had increased to 75. 30 million people were older than 65, 12% of the population, and many were healthy. Now, in fact, there are centenarians, over probably 100,000 centenarians, people over 100 in the U.S. And some are healthy, but many are not. There are a whole bunch of issues as we start to get into the 90s and 100s. But here's a really interesting and striking possibility that was published in Lancet a couple of years ago. As of 2000, 50% of those born that year are expected to live to be 100 years. These are mostly based on European, North American, Australian statistics, but it's a pretty sobering thought, because age rivals all other risk factors for the common forms of Alzheimer's disease and stroke, and these are the diseases that are turning into the tsunami of the next 50 years. Because this is what's happening. We're seeing the inversion of the usual population distribution, where youth support the elderly, to where more and more there are going to be elderly and fewer youth. 10,000 baby boomers are reaching 65 every day in the US. And for the first time in history, this is another factoid I thought that was very interesting, in developed and some developing countries, adults will have more parents than children. Just think about that. So we've had some figures about dementia prevalence in Canada, and these numbers are even a few years out of date. It's about 500,000. It's going to double, a little more than double in a generation. Economic burden is currently about 15 billion. These are out of date for a few years. You can add inflation to that. About 153 billion in a generation. The economic burden is expected to reach 872 billion cumulatively over the next 3 years, just in Canada, which is population-wise the size of California. So you can see that this is a major threat to the future of mankind. Now, in fact, the facts seem to show that women tend to live longer than men. At most ages, men die at a higher rate from numerous causes. And this is just showing some examples. These are based on some studies that have been done and have looked at different countries. And you can see, for example, that heart disease is a major issue in men. It still remains higher in men, but it goes down a little bit and starts to approach women as time goes on. Cancer is a little more kind of egalitarian. Heart disease, higher in men, goes down. Flu and pneumonia, sort of comparable. Strokes actually go up in men toward middle age, and then women through survival start to have more strokes. And then chronic obstructive lung disease affects men more than women, but with younger women smoking more, that may be a different future. But that just shows you some of the comparisons of these different diseases. So why do women live longer? We don't really know. There are various hypotheses, such as immune functioning might be more active in women. Women have more autoimmune diseases, for example. Estrogen actually has protective effects until you lose it post-menopause, right? There may be compensatory effects of having extra chromosome material on the X chromosome. So women have more genetic material, interestingly. Various hormones, besides just estrogen, are a little different in men and women. And also there may be differences in something called oxidative stress, which is an aging-related change that happens in the body's ability, in the cells and all different parts of the body to keep reproducing or keep surviving well. So the paradox is that although the mortality rates are actually lower in women, they live longer at virtually every age, women have a higher overall rate of physical illness. More disability, more doctor's visits, more hospital stays, more chronic disease illnesses, and therefore maybe more suffering in some way, more long-term suffering, arthritis, and so on. So that's kind of interesting. And why is that? We don't understand that. What can we do about it? Dementia incidence. Now, incidence means the number of new cases per year, okay, or per 1,000 person years, they often call it. And in generally speaking, the incidence rates of dementia and Alzheimer's disease are higher in women, especially at old age, because by then there's a very high rate. Maybe 50% of people will have Alzheimer's disease present when they're in their mid-90s, for example. And here's one study where over a 3-year period in elder people over 65, women had 42 versus 29 per 1,000 person years incidence of development of new dementia. And overall, the risk of getting Alzheimer's disease is about almost twice for women as it is in men. And these are just some of the data that supports this. However, it's not so straightforward because actually some of these numbers are driven more by European statistics. This is showing this big difference in the incidence rates of Alzheimer's disease, contrasting the American European studies. In the European studies, a lot more than in the American studies. You can see that they're closer together. Even that was surprising to me, and I don't think has been sufficiently explored. Maybe it has to do with where the populations were studied. In the Framingham study, a very famous study, that's a suburban area in Massachusetts, and people are probably well cared for, generally speaking, in terms of their health care. Maybe that makes some sort of difference. We know that vascular disease and vascular risk factors, sedentary lifestyle, for example, are risk factors for developing faster brain aging and Alzheimer's disease. So with respect to genetic factors, there's also a risk factor for Alzheimer's disease that's very important. It's called the apolipoprotein E4. Some of you may have heard about it. It increases the likelihood that you will get Alzheimer's disease. And if you have an E4 gene from your mother and your father, you have a double E4, you've got a very high risk of developing Alzheimer's disease. It's not used as a genetic test because you don't have to have the E4 in order to become demented or have Alzheimer's disease. But if you have it, you're at greater risk, and it may start at a younger age. Now, by dementia, what we mean is a person has developed cognitive problems, memory problems, thinking problems that are sufficient to interfere with their independence, their total autonomy, looking after themselves and living in society on their own. That's really what we mean by dementia. It's a kind of a term that's used to indicate a certain severity that gets reached in cognitive abilities that mean the person needs help. That's really what it means. Because a lot of people wonder what's the difference between dementia and Alzheimer's disease. Dementia, there may be 80 causes of dementia. Alzheimer's disease is one of the most common. So in the total population, the E4 individuals are more likely to get Alzheimer's disease, but there's a stronger relationship about the E4 status in women compared to men. So if you are E4 positive, you have 4 times the risk, whereas you have twice the risk of this being the case if you're a man. Usually this is just expressed in population terms. It comes out roughly, a little over 2 times the risk if you have one E4 gene. But when you break it down by men and women, there's something really interesting going on there that we don't understand yet. And this is just shown here where you can see that if you're E4 positive, you're more likely to become demented faster if you're a woman than if you're a man. These are the kinds of, this is the kind of evidence that's out there that we're not sufficiently studying. This is just the point about the, it's not all hormones, about the fact that women have more chromosome material than men. So that's turned out to perhaps make the female sex hardier and sort of more able to withstand chronic illness and survive, but at the same time be much more subject to these diseases, as I mentioned. And we have no idea what that is. What's on these chromosomes? Now we're getting to a point where we can begin to identify all the genes on these chromosomes, and we can begin to understand some of these differences, but we're not there yet. There may also be differences that relate to receptors in the brain, now getting into the brain itself, which we don't understand. We know that overall, brain volume is bigger in men. Just look around, there are few brave men's souls who are here today. But generally speaking, men are bigger than women. That's why we have different sports, except maybe hockey. I don't know, we're trying to, field hockey perhaps. But however, when you control for the total size of the head, women actually have more brain. They actually have more gray matter, the important part of the brain. Right? Blood flow is higher in women, and we know that estrogen increases and improves blood flow in women. Estrogen has a protective effect against stroke and heart attack in into midlife, and then once you become postmenopausal, you revert more to the male situation. But there's a really intriguing and interesting trick that nature has played on us that most people aren't aware of, and that is that testosterone gets converted to estrogen in the brain. So men continue to make estrogen in the brain until they're in their 80s and 90s, whereas women go into estrogen withdrawal. And that has consequences. Some of these differences in some of the neurotransmitters, I just mentioned this one, which is the receptors that are related to mood. They're called serotonin receptors, and women have more of them in their cells in the brain generally than men do. Does that explain why women may be more susceptible to mood disorders? We don't know. But these are fascinating observations that haven't really been put into a coherent understanding of why this happens, and also what we might be able to do to anticipate and treat these conditions. So now let's get to the brain itself. What I'm going to do now is talk to you and do a quick kind of anatomy lesson and just run you through the kind of work that we do trying to understand brain changes in Alzheimer's disease and in normal aging. So this is going to be a kind of a quick education for all of you who don't look at brains every day. But I do as a neurologist and also in the research that I do. And the first thing to me, it's a work of beauty. I get joy just seeing how amazing this organ is that really is what drives everything we do. And I just want to show you a typical slice. This is a slice of the brain, sort of in this direction. And it shows you the gray matter, the cortex, which is actually orange here. It is actually gray on an MRI scan. This is an anatomical MRI scan. They're getting better and better, and we can actually measure this. We can actually measure the cortex. That was not possible 15 years ago, and we're getting better and better at it. And we can do it in an automatic way that didn't used to even be possible. We can send an image of a brain, if it's done in the right way, off for processing overnight, and it'll come back and it'll tell you what's the thickness of the cortex in certain regions. So there are computational methods now that allow us to do things that we never could do even 5 years ago. So that's what the gray matter looks like. And this is what the white matter looks like. We've made it green just so it'll jump out at you. But it does look white on this type of scan. This is called a T1 scan, a T1 weighted scan. It brings out the anatomy of the brain, and we do it in 3 dimensions. So we have a whole brain all that can be looked at in different directions. And this is the white matter. So this is the part of the brain. This is the part of the brain that connects the gray matter together, and the top to the down, and the front to the back, and the side to the side. This is the white matter. These are the fibers of the brain. This is the part of the brain, for example, that gets affected in multiple sclerosis. It's the white matter. This is the part of the brain that changes with aging, and you start to get what we call white matter disease, and I'm going to show you examples of that, more in women than in men, actually. So if you're going to start doing brain anatomy, though, the first thing that you have to understand is that bigger is not necessarily better. Otherwise, there really would be a great disadvantage to the female sex. And in fact, in the 19th century, when people started measuring head size, there was biological evidence for why women were inferior, because their brains are smaller. And as an example of how bad we are at actually investigating this, interesting phenomenon, how can on average, women are generally as smart as men. There are some sex differences in abilities. Women tend to be verbally, as a sex, more advantaged. Men tend to be visually, spatially more advantaged, probably for evolutionary reasons we don't really understand. But in the 19th century, it was, there was obvious reason for why women were inferior, shouldn't have the vote, and all the other things that come along with that, right? And believe it or not, it wasn't until the 1990s that a scientist actually asked questions about that. Like, how can that be? How can you have equal ability in a smaller amount of material? And that is actually Sandra Whittleson, who's a researcher in Hamilton, who's been very interested in sex differences. And I won't go into it, but the long and short of it is she was particularly interested in why are women generally better at language function, and yet they have smaller brains. And we know that the part of the brain that's really important for language is on the left side of the brain. It's called the temporal lobe. So how can that be? It's smaller. So in fact, what it turns out to be, when they did cell counts on young men and women who had cancer, who knew they were going to die, and agreed to donate their brains and be studied before death, and then have their brains examined post-mortem. What she found was that in these crucial areas, the packing cell density, the number of cells and connections in women is more efficiently organized. So you actually have the same amount of kind of communication, connectivity power in a smaller amount of real estate. So it's very interesting. That took till the 1990s. And there was anatomical methods that were available from the 1900s. So it just sort of shows, I think, part of the difficulty we do face, which is we don't sufficiently investigate this in the human condition. So because of that, because of these differences in size, the fact that you have to take into account these differences in head size, you have to make corrections for head size. And a lot of the brain imaging work doesn't do that. So the same amount of a volume in a smaller head is a relatively bigger proportion than in a bigger head. And so you can't just look at a volume without contextualizing it in terms of the size of that head. And the idea is that everybody's head and their brain are nicely and snugly fitting together. Okay, so you fit into the skull in proportion to your head size. And so if your brain starts to shrink, what happens is that some of this turns into just spinal fluid. The dark part of this is spinal fluid. So you get more spinal fluid and you get less brain. Does everybody get that idea? And so you have to go through extraordinary measures in order to, you start with a brain, with a skull and everything there, to strip away the non-brain parts, but preserve the spinal fluid parts so that you can actually truly say how much brain is there in relation to what it should be. So that has not been done by a lot of anatomical studies. It takes extra time, it takes extra processing, but now thanks to various methods, it's getting easier to do this, and it's something that our group has been particularly concerned about, because you have to correct for the starting size of the brain before you make any measures between people. And this shows you, if you strip away the brain, which a lot of people do, and then they use the brain as its own denominator, you're going to miss a lot of the shrinkage. This is showing the spinal fluid surrounding the brain. It cushions the brain, and as the brain shrinks, there's more of it. So you need to know what proportion of that intracranial capacity is actually brain. It's called the brain parenchymal fraction. And that's because parenchyma just means the brain itself. And this is just this idea. This gives you, group average for brain is about 900 cubic centimeters. That's like, we can now say that. About 75% of that is brain itself as opposed to the CSF. And this shows you an example of someone whose average brain is fine. In fact, it's 941 cubic centimeters, this little red dot here in a scatter plot. This is from some of our own data. But the brain parenchymal fraction is only 64%. So that person has atrophy, has shrinkage. Atrophy is a technical term for shrinkage. And this is where it starts to get really interesting, because here we have men and women, And what you'll see is that generally speaking, this is the kind of average of all of these, the trend, women tend to have bigger brains than men. Brain parenchymal fraction. This has also been done in some larger studies where they had hundreds of people and they had children and parents, and they follow them. And what they showed that is in middle age, there's not so much difference, but as you get older, men's brains shrink faster than women's. These are normal people, no complaints in the community in Framingham. So in the Sunnybrook Dementia Study, which we've been conducting for a number of years, we have been doing comparisons between people who have Alzheimer's disease, people who have no problems, who volunteered to be normal controls in our studies. And this just sort of shows you some analysis that we've done specifically for this talk, where we were looking at, these are the average ages of the individuals here. Education, we're relatively well educated, but usually controls are a bit better educated because they're volunteer retiree folks who give of themselves to help us learn about what's normal. Now, they're well educated, so they're probably a little above normal in some respects, because education, as well as age, can actually affect brain volumes. Maybe it's through brain connectivity. It makes the brain have more reserve against the attacks of various kinds of diseases, such as Alzheimer's disease. So we have these pipelines, and I'm just going to show you some data now. So you've already heard me talk about white matter and gray matter, and so this is comparing Alzheimer patients, men and women. These are mild, moderate stage Alzheimer patients. The white matter is not that much different in men and women, either normal or in Alzheimer's disease. But even in our small sample, we see that the women have a little more gray matter if they have Alzheimer's disease, and also normal controls. So we're finding the same thing that has been reported, but now it's in a specific series. There are other ways that we can measure aspects of the brain. These are the ventricles. This is where the spinal fluid is produced, and then it circulates around in that sort of CSF space. It cushions the brain. So if you shake your head, it's cushioning you, and you don't get a concussion just from shaking your head. But if you're playing hockey and you get hit in the head, then you can get a concussion. So what we do is we can measure this separately, because it turns out that this is a very good measure that summates all of the processes going on in the brain that can end up making it enlarge as you get older. So this is a younger person's brain. As you get older, your ventricles enlarge. You get a bigger space there, just as you get more space around, because the brain is shrinking. Unfortunately, it starts to do so around age 50, men and women. And it keeps on going little by little. There's a certain percentage per year. So this is an example of a normal elderly person, maybe in the 70s, and this is what their ventricles. This is now a three-dimensional reconstruction of this. Like, you can take this 3D structure called the ventricles, and this is actually what it looks like. We're kind of looking down on it, so you can imagine it's a three-dimensional structure. You can see that there is a little shrinkage even over 1 year. I mean, in the sense that it's getting bigger because the brain is shrinking. But here's an Alzheimer patient. You can see already that it's much bigger, this ventricle, it's expanded, and it gets larger even faster over time. So the brain is really shrinking fast in Alzheimer's disease. So just as is the case with the gray matter, it turns out that in Alzheimer's disease, men's ventricle size gets bigger faster, and this is after we correct for the head size differences, right? Whereas it's not so different in normal controls. This area of the brain is really important. This is the hippocampus. This is the part of the brain that helps you learn new information. This is what gets attacked early on in Alzheimer's disease and starts to take away the person's short-term memory. Vital structure deep in the brain, and it is the one that starts to shrink first in Alzheimer's disease. The other thing most people don't realize is that Alzheimer's disease is incubating in the brain over many years, maybe 20, 30 years. This is becoming important because it may be an opportunity to intervene 10 or 20 years before the person would be expected to show the disease. So it's a huge opportunity for prevention and possibly even for intervention. We can talk about that later if we have any time. So we now have methods that can automatically we call it segment, automatically take out the hippocampus and look at its volume so that we can then see, is it shrunken? Is it okay for age? I mean, I imagine that in a few years, you can probably get a hippocampal volume that's just like having a blood value for your hemoglobin. This is within, this is 1 to 2 standard deviations below average for age, right? This is going to happen because this can be done in 2 hours of off-site parallel processing. It would take you about 4 hours to trace that painfully, right? But now that that's been done and you have templates and you have machines that can go and do matching to templates, you can send this off to a computer and it will overnight do a whole bunch of these, and then it'll spit out what the volumes are. So what about the hippocampus in the context of Alzheimer's? Well, this is why you really have to do the correction for volumes. Okay, so it looks like in this case, men have, in Alzheimer's disease, have bigger hippocampi than do women, and also normal controls. But look what happens when you correct for head size. First of all, not really different in men and women. And in fact, women have more hippocampus relative to men who are normal controls. In the case of Alzheimer's disease, the disease is attacking it, and it's sort of ruthlessly attacking no matter what your sex is. But in the case of the hippocampus, are both, by the time they come to us, they're both, in both men and women, the hippocampus has been shrunken. Finally, I just want to end with something called small vessel disease, which is how little strokes can affect the brain, and also there are interesting differences in men and women. So this is an example of a little stroke deep in the brain in an area called the thalamus, and this just shows, we can actually get its volume, and we know, it's kind of a half cubic centimeter, something like that. Even smaller ones can be seen in people. These are like 3 millimeter little holes in the brain. They're called silent strokes because the person doesn't know they've had them. It's just when you do a scan, you see them. And they look like bright signals on a different type of acquisition that you do at the same time as you do this kind of scan in an MRI. So they're called silent strokes because they're little tiny ones, and they're just noticed in population studies. They've shown up. But they're not benign. And in any of the studies that have actually followed people, it turns out if you have silent strokes, this was in the Rotterdam study where 1,000 people were followed over 4 years, you were more likely to have a cognitive decline, you had twice the risk of becoming demented, and you had 5 times the risk of stroke. 3 times even after you corrected for high blood pressure and other things. So in other words, they were telling us that it's an active disease of some sort that we need to understand. It tends to be more commonly seen, these little silent strokes, in people who have high blood pressure, people who have diabetes, people who have cholesterol problems, people who are sedentary, probably, all of those things that we know as vascular risk factors. And here again, at least in some series, it looks like women tend to have more of these than men. This is the prevalence of them, and this is if you follow them over time, you see more of them developing. Okay, so you can just see by decade. So it's small vessel disease seems to be something that women are more subject to. And this is just in our own series, which basically shows the same thing. This is not significant, but in Alzheimer's patients, we found that there were more of these silent strokes in the women. There's something else called white matter disease, which is a great interest of our group. This refers to normal controls, vascular cognitive impairment, not dementia. This is something like mild cognitive impairment. And this is Alzheimer's with cerebral vascular disease, showing something that we see as people get older. And this is a severe amount of it. It's present in about 20% of people over age 65. And it's associated with problems with walking and cognition. But it happens and develops gradually. We don't exactly know why. We think it's to do with venous disease deep in the brain. This is what it looks like on a CT scan where it's dark. And this is in another example. And in fact, some degree of this, whether it's just little dots like this, is pretty frightening. It's present in 95% of people over 65. So maybe not too many of you over 65 like some of us in the room, but it's a very common pathology of aging, of human aging. And when you have extensive disease, such as the examples I showed, then you are slower, you have gait difficulties, you fall, And there may be a threshold effect. Now, again, this is not benign, because if you have that, you have a greater risk of stroke, you have a greater risk of dementia and death, and you have a greater risk of decline in your mental abilities. So this is just showing you the way that we can do a very comprehensive analysis of each person's brain to know exactly how much gray matter, white matter, and all these different things that they have. And I'm not going to dwell on that. It's just to show that over time, we're showing that even over a year or two, we're showing that the women are losing brain less quickly than men. But the paradox in all of this is that for the same amount of brain, they show more cognitive impairment. So in fact, that is one of the most intriguing paradox. It's a bit like the mortality-morbidity paradox, where women live longer, but they have more chronic illness. Women have more brain, but for the same amount of brain, they have the same degree of cognitive impairment. So what is going on there? Is there something missing? Is it lack of estrogen? Is it lack of some other chemical ability that we just don't understand? So that is one of the most fascinating paradoxes that we have in the consistency that's coming up from the various types of literature. This just shows that men have more progression of those lacunar strokes than do women, although women have more of them in general. So I'm going to finish. I'm sorry I've gone over time, and I appreciate those of you who are still staying. I just want to come back to say that it is complex. I hope you appreciate that now, that there are a lot of niceties in how we do these measurements. This is not you just go out and kind of measure a brain. This is, you got to think of all these ways of making sure you're getting the brain inside the skull, and you're taking into account the amount of shrinkage in different parts of the brain, different diseases. Frontotemporal disease, frontotemporal degeneration affects different parts of the brain than Alzheimer's disease, and we see these patterns. Genetic factors, hormones, immune reactivity, social environment factors all probably play a role we don't fully understand. We can now measure it. We're finding intriguing differences, especially at older age and with diseases like Alzheimer's disease. So the bottom line is we do need more research, and we do need advocacy for the gender lens and for concentrating on both men and women, whatever the animal may be, human or rodent. And happily, in the Canadian Consortium for Neurodegeneration and Aging, there is this cross-cutting program core theme, which is going to make everybody look at these differences. Because in that project, there will be thousands of people followed over several years from different disease states and with different kinds of interventions. And it will be really important to have this kind of information. So I just want to acknowledge many sources of support, including philanthropy, that's helped my lab and various agencies, and the many collaborators that really make this all work. I apologize for going over time. Once I get going, I can't. Thank you.